Hello students, welcome again. Welcome to my channel. Okay, so for the NEET examination, I have come upon with a series. In this series, we will be discussing some questions which are important, obviously, for your NEET examination. So let's start with the first question. The first question is, which one of the following is not a criterion for classification of fungi? Okay, so we all know fungi, that fungi cannot produce their own food, that is, they are heterotrophic. Okay, and they derive their nutrition from what? From dead and decaying organism. Okay, so each and every fungi is having that criteria, that is they are deriving the food from dead and decaying organism. Okay, so let's see the option. The options are mode of spore formation, fruiting body, morphology of mycelium, and mode of nutrition. Okay, so the first option, what is it telling? Now, mode of spore formation, there are different modes of spore formation in different groups of fungi. Okay, they produce basidiospores, they produce conidiospores, like that. Fruiting body is not also the answer. It is asking for which one is not the criteria. The mode of nutrition will be the answer. Okay, as because all of the fungus are having heterotrophic mode of nutrition, they are dependent on dead and decaying organisms on their nutrition. Okay, now let's go to the second question. Okay, the second question is the match the following question. Okay, so in this type of questions, in match the following, you will just try to make one or two options correct, and then you will try the elimination procedure. Okay, so I will teach you. Acha, before coming to the answer, just pause and try to give the answer yourself. Okay, so the first one is GLUT4. So what is GLUT4? GLUT4 is something which is a transport protein. Remember it. It is a transport protein. Okay. It helps in transferring what? Glucose inside the cells. Okay, so without this GLUT4, glucose cannot go inside your cell. Okay, so GLUT4 will be matching with number 4. See, where is D number 4? See, A and 4. Where is it? Only in one options. By just knowing one answer, you have done it correctly. Now, second one, see, insulin. Insulin is what? An enzyme or an hormone. See, insulin will be a hormone. Yes, many people who are diabetic, they take insulin, right? So B1A4 and C2 is the enzyme. Okay, and C will be enzyme. Trypsin is a enzyme. Okay, it is a protein cleaving enzyme. Remember, trypsin is what? Trypsin is a protein cleaving enzyme, which means it cuts the protein. Suppose this is a protein, it will cut the protein. Okay. Now D is collagen. Okay. D is collagen. D3, that is intercellular ground substance. Collagen is intercellular ground substance. Okay. Means inside the cell there is a ground substance that is known as collagen. Clear? So C will be your correct option. Now comes the third question. It is a very easy and direct question. So you have read about Agrobacterium tumefaciens. Okay. It is a bacteria. We study it in the biotechnology chapter. So inside that bacteria there is a plasmid that is known as TI plasmid. What is that? It is the tumor inducing plasmid. What happens if this Agrobacterium attacks what? Attacks a plant? Gall disease, gall like structures that are what? That are tumors. Tumors will be produced. So the correct answer will be tumor inducing plasmid. Okay. Now come to the next question. Okay. Yes. See, there are given two statements. First statement says mitochondria and chloroplasts are both double membrane bound organelles. Yes, very correct. Mitochondria and chloroplasts are double membrane bound organelles. Remember it. These are double membrane and autonomous organelles. Why? Because they are self-replicating. They have their own DNA. Okay. Mitochondria and chloroplast are having their own DNA. Now the statement to inner membrane of mitochondria is relatively less permeable as compared to chloroplast. No. That is totally incorrect. The answer will be the first one. Statement 1 is correct, but statement 2 is incorrect. Okay. First one will be the answer. Now comes, this one is a very easy question, but you have to remember the list. Okay, remember the things to answer this question. The question says following are the stages of cell division. Obviously, what are the stages of cell division? We know there is interphase. Okay, and there is what? Mitotic phase. Okay, so this is the cycle. There is interphase and mitotic phase, and in interphase there is G1, S and G2 phase. Okay, G1, S and G2 phase. So the first stage is what? The gap 1 phase. This one is the first stage. So it will be what? E will come first. This one is the first. Then comes the S phase or the synthetic phase. This is the second one. Then comes the G2 phase. This one is the third one. Then what? The nucleus division will happen. That is the karyokinesis. Okay. Which one is this is the fourth one. So see, and obviously this will be the fifth one. What will be the answer now? What is the correct sequence? They have asked the correct sequence. Okay. So obviously, see, first there are two options. By elimination method you could have gone also. ECADB. E, then what? The second is C. Obviously B will be your correct answer. Okay. So this is the approach you will have to do in the exam. This should be your approach. Which questions to attempt, which questions how will you deal? Okay, you will have to know, and that comes with practice and a lot of practice. Okay, so the next question, it is a bit hard because you will have to remember the total molecular biology. Each and every enzyme name you will have to remember it. See, but you must try what the elimination method, what I told. See, the first Tata box, you know what Tata box is present in the promoter. So D2 Tata box, you know that to start the transcription process, the Tata box is required. Okay, the polymer is binds to the Tata box. See, D2, where is D2? This and this, these two options are D2. Then see, termination of transcription. You would have known that termination sequence of termination of transcription occurs because of the row factor. There is a factor named row factor. Okay, the row factor, when this term row factor comes, you must remember the termination of transcription will occur. Okay, okay, so B3 will be the answer. B3 will be the answer. See, where is B3 present? In this option, D2 and B3. Obviously, that would be your answer. And see, splicing of exons. Splicing of exons are done by snRNPs. C1. Okay, short nuclear RNPs. And RNA polymerase produces tRNA and snRNAs. Okay. Now comes the seventh question. This has came from your experiments portion. Okay, you have learned about the semi-conservative method of DNA replication, where suppose this is a DNA strand. When a new DNA is produced, one strand will be the old and one will be the new. Okay, one will be the old strand, one will be the new strand. That was proved by whom? By Michelson and Stoll. Okay, that is the semi-conservative mode of replication. See D2, where the option D2 is there? Oh, there. See, such easy question. Only by doing one option you will get it right. Hargobind Khorana, this was an Indian scientist who invented or discovered the genetic code. Okay, and Griffith did the transformation experiment with the mice. Okay, and Jacob and Monod, when you see this Jacob and Monod name, these two scientists worked with lactose operon or lac operon. Okay, so answer will be D. Now the eighth question. See, a definition has come. Okay, so the definition is capacity to generate a whole plant. Suppose there is one cell. This one cell generates the total plant. From this one cell, a whole plant is generated. Obviously, the cell divides. There is differentiation. The organs are produced. That are different things. But the capacity of the plant or the whole plant from any cell to make a whole plant is known as totipotency. Remember it. This feature is found exclusively in plant but not animals. Suppose if I cut my hand, can a new me be produced from the cut portion? No, it is not possible. Okay, so what we will have to do? In case of plants, we just take any portion of it, and from that any portion the whole plant is produced. That is known as the totipotency. Okay. Acha. This is from your ecology portion. An equation is given, dn by dt equals to rn and k minus n by k. So what does the k indicate? K indicates the carrying capacity. It is just a formula based question. So if you ask me what is the carrying capacity is nothing but how much a environment can carry. That means suppose in your home, suppose this is your home. Oh, sorry. Suppose this is your home. Okay. There is capacity for 5 people to eat. Okay. And if 7 people come, there will be what? Competition. That is not possible. Okay. So that is the thing. Now, the last question. As I told you earlier, you must just pause and then watch the videos. Okay. And then try first. And then watch the solution, not the video. Okay. So, this is another match the following question. I have taken more match the following question. Why? Because in this match the following question, more informations can be gathered in a very short span of time. You all try to do maximum of match the following questions. Many informations you will be getting. Okay. Now see, citric acid cycle, where it occurs. Glycolysis, where it occurs. Glycolysis occurs in the cytoplasm. Suppose this is a cell. This is its nucleus. This is its mitochondria. Okay. Sorry, my mitochondria has become very bad. This is the mitochondria. This is the nucleus. Glycolysis occurs in the cytoplasm. Okay. So B1, where is B1? See, again same thing. B1 is present in only this option. Questions are not that much hard. Okay. Now citric acid cycle, it occurs in the intermembrane space of mitochondria. Okay. See, suppose this is a mitochondria. Okay. This space is known as the intermembrane space. And citric acid cycle occurs there. Okay. And see, now the electron transport occurs in the inner mitochondrial membrane. This is the inner mitochondrial membrane. Inner. Okay. Electron transport occur in the inner mitochondrial membrane. And D is 3. Intermembrane space. Okay. So, thank you for watching. Have a nice time. Thank you.